Because I see my PowerPoint loaded on the screen, and
you see that we don't see that.
Oh wow!
It say that you are started sharing, but
okay. Now I see it.
Yeah.
No, I see. Yes, I see Francesco Caldarelli, and you are in the. So that's much better. Okay. Now it's okay.
So you should see the first page of the PowerPoint with the title.
Yes, it is okay.
Okay.
So we are a little bit early in the agenda, so just wait a couple of minutes.
Well, I was in the breakout room, and then it says exit from the breakout room. Okay.
Just to be sure, if I move to the second slide, you see in real time. I'm moving now.
No, nothing has moved.
I think there's a bit of a lag, yeah.
Wow, that's a problem.
Nothing has moved yet.
Yep.
Now,
okay, now yes.
Oh, there's a lag. I don't know why.
Well, actually, it was about 5-10 seconds.
So a phase shift. Correct it with phasers, I think.
Yeah, advance the phase.
I hope this is not an issue. I try to speak slower. And
well, anyway, I don't think at this point there is nothing you can do. Well, other than to give the presentation to Lionel or whoever, and then they will advance. But that is very complicated. So don't try to go up and down a slide.
Yeah, sure. Okay, I'll try.
Don't worry, I will. Yeah, I think it's gonna be okay. No, don't worry. We will basically turn off all our cameras just to save bandwidth, and maybe this help.
Okay. Because obviously I'm at home and the connection is not the one I have in the lab, but this time I cannot be in the lab.
Okay. So anyway, look, until now the meeting has been working very well, but
yeah, don't worry.
So we have some problems of zooming and so on, but anyway.
So okay, I'll introduce you, Francesco. Well, it's my pleasure to introduce Dr. Francesco Caldarelli, Associate Professor at the Applied Physics Department at the Laboratory of National Enterprise for Nanoscience and Nanotechnology, NEST, in the Scuola Normale Superiore Pisa, Italy. Francesco is a biophysicist with a strong background in biology, and I will say that in his first career, he addressed many biological questions with all these new techniques developed at LFT, like FCS, FLIM, and he has done major contribution of the development and application of IMSD for diffusion within in vivo systems. He has a really strong record of publication and in different subjects. I always admire the capability to address different subjects that Francesco has. And this allow him to got a prestigious grant, which is the RCR grant from Europe. I am a big admirer, as I say, of his career, and I always find very nutritive and very stimulating our discussion about any kind of topics, because I always enjoy discussing biology as well as fluorescence or instrumentation with him. So thank you, Terco. I pass the word to you.
Thank you very much, Lionel, for your very kind introduction. Good afternoon to everybody. So as I was saying, if there's something good in these bad pandemics is that we can get in touch virtually, even if we are far from each other. So I'm very glad to be here and to have this opportunity to talk to you. So going straight to the topic of my application talk, I would like to start by using the words of a physicist, Erwin Schrödinger. We are in 1944, and he's wondering about the complexity of life as the result of many single molecule events taking place in space and time within the boundary of a living system. Things are getting complex. However, if we think how to grab these events, that is a common task for biophysicists, is our task every day. We could be optimistic if we think that nowadays we are able to grab the position in space and time of even single molecules, as I hope you are going to see in this movie and show you. Well, there's a technical limitation in doing this. And the limitation is linked to the fact we decided to localize the molecule. And localization is a time-consuming process. Still, you can get in acquisitions like these information about the aggregation rate, the number, the diffusivity, and you get the trajectory because you are localizing the position of the molecule, and you get a complete spatiotemporal description of the molecular behavior, the so-called diffusion law. But as I was mentioning, you have a technical limit linked to the decision to localize. This is time-consuming, and so there's a limit in the temporal resolution achievable. In other words, not always acquisitions like these are feasible technically, experimentally. Can we bypass this limit? Well, the answer is yes. What happens, for instance, if we decide not to localize, but just to detect, to record the signature of the passage of the molecules across our observation volume, for instance, the laser spot. So in your measurement, you get many of these signatures, many of these fluorescence fluctuations, You end up with a plot of fluorescence fluctuations in time, and with the mathematics, you know very well that is known since more than 100 years and applied to fluorescence since 1972, the seminal paper by Magdelson and Webb. You extract the G of tau, you extract again information about the aggregation state, the number, the diffusivity of the molecule. But obviously, you don't have information about the position of the single molecules, you don't have trajectories, you cannot achieve a complete spatiotemporal view on process. So the question is, can we bypass even this limit? Well, the answer is again, yes. The opportunity is given by a huge number of spatiotemporal variants of the basic concept I showed you of fluorescence fluctuation in a point. Many techniques flourished and were proposed in the last, let's say, 15 years. And our starting point to generate the IMST technique that I'm going to show you today is one of these techniques, STICS, spatiotemporal image correlation spectroscopy, proposed by the Weizmann group in 2005. And the basic concept is very easy to explain. You start with a spatiotemporal acquisition of fluorescence fluctuations again, and you start comparing each acquisition, each image in your stack with itself. Obviously, nothing is moving if you compare each image with itself. And with the mathematics that you see here below, that is very similar to the basic G of tau applied to images, you get, if you compare each image with itself, the spatial correlation of the objects. Then you introduce the time delay between the images, and you start comparing each image with the following one in time. And assuming molecules are diffusing isotropically, they will change position in one image with respect to the other. And your calculation, the spatiotemporal at this point, correlation function, will depict that average behavior of the molecules decreasing in age and increasing in width. And you do this calculation for all the possible tau delays you were able to measure, to grab in your measurement, and your spatiotemporal correlation function will continue to depict this behavior by decreasing in age and increasing in width. So far, so good. This is STICS. Then you introduce fitting, and the most appropriate fitting we can think of is Gaussian fitting. And the variance you see here highlighted of cushion fitting is a quantitative estimate of the average displacement of the molecules, because it's an estimate width of the spatiotemporal correlation function. If you plot that quantity as a function of the tau delay, of time, you end up with the so-called IMSD plot, a plot of the mean square displacement, average mean square displacement of the molecules derived directly from imaging, with very peculiar properties. No need for complex labeling. It works well with dim molecules because, remember, you decided not to localize. So you're just measuring fluctuations, and even dim fluctuations are okay. Large amount of information in a single measurement. No need to pass through localization, to pass through trajectories. Information on the size of the diffusing object, and I will go to this point in a moment, compatible with any kind of spatiotemporal imaging, because I talk about images, but it could be lines. Any to simple space and time is okay. Unprecedented temporal resolution down to microseconds, again, because you decided not to localize. So we tested first the properties of the IMSD analysis by simulations. Suppose you have a box with a particle diffusing, and you measure your spatiotemporal correlation function. You analyzed by Gaussian, you extract the variance, you plot the variance. If the molecule is diffusing isotropically, you will get a straight line, and the slope of the straight line will be proportional to the diffusivity of the molecule. That's obvious, and this is exactly what we expected to be. Again, it is expected, and the simulation is telling us that if you put the molecule in a mesh of not penetrable barriers, you will get an IMSD, with the shape of confined diffusion, and a plateau value whose height is proportional to the size of the mesh, is giving you information on the size of the mesh. Again, so far so good. If your barriers, instead of impenetrable, cannot be overcome by the molecules with a certain probability, you will end up with a high MSD plot that is giving you two regimes of diffusion you're going to see in a moment. One, a very short time that reflects the diffusion within the mesh, and the second one, a longer times that reflects the probability of the molecules to overcome the barriers, and the slope of the second regime obviously will depend on that probability. So I promise to comment a little bit more on the size of the diffusing object. Yes, the IMSD analysis is giving you not only dynamic information, information on dynamics of molecules, but also some kind of structural information in terms of size of the diffusing object. Because if the size is that of a point-like particle, few nanometers, let's say below 20, 30 nanometers, you will end up with an intercept of the y-axis that is close or coincident with your observation volume. What's the meaning of that intercept? Well, philosophically speaking, is displacement at time zero. Displacement of a molecule at time zero is the molecule itself. If the molecule is a point-like particle, as I mentioned, you get your observation spots. But if your molecule has a sizable, a measurable size above few tens of nanometers, you can estimate that size because you will get an intercept that is higher than the one that you can calibrate of your observation spot. So you have also this possibility within this technique to get this kind of information. So these are in silico tests of the validity of the analysis. Let's go to real applications. So let's skip this slide to go to the first real application I want to show you. That is on membrane structural organization. you know that in the last 40 years, our concept, our comprehension of structural organization of the membrane has changed a lot, from the fluid mosaic model, 1972, Singer and Nicholson, to the current view based on Kusumi and many other researcher work of the so-called fence and picket model. So molecules are confined by the active meshwork and are also transiently confined within the so-called lipid rafts. So the things are more complex than expected based on the previous works. Can we measure by the IMSD this complexity? Well, first of all, let's compare to the single particle tracking people, because the people in the field started using single particle tracking and started measuring trajectories. One was Kusumi, as I mentioned. And if you take some of these molecules, for instance, the transferrin receptor, you will get a very nice measurement of transient confinement of these molecules. Transient confinement by what? By the active meshwork, I repeat. So you label your receptor, as in the drawing here, with organic dyes, GFPs, quantum dots, obviously. As mentioned before, the brighter, the better. You track position of isolated single molecules, you get a trajectory, you analyze the trajectory, you get the standard mean square displacement, and the shape of the mean square displacement, as you can see here, is that of transient confined. And you can draw the mesh, as you can see on the right. And you can also get an estimate of the mesh size. The mesh is imparted by the actin filaments of about 500 nanometers. These numbers are measured from a position with a temporal resolution of 10 to 20 milliseconds, okay, of single particle tracking. But if you get the mesh size by electron microscopy, you get an apparent inconsistency of data, because the mesh size should be much less than 500 nanometers. Can we recombine these apparent discrepancies? Well, we address this topic, you may expect, by increasing a lot the temporal resolution, because deciding not to localize, we can go much faster than 10 milliseconds. Actually, we use temporal resolution down to 100 microseconds, either by total internal reflection, fluorescence, and camera-based imaging, but also these speeds can be reached by raster scan, okay? For instance, along lines with a raster confocal laser scanning microscopy. So both cases, you reach 100, 120, 150 microseconds temporal resolution. So orders of magnitude higher, faster than single particle tracking. So this is the plot we got with transferrin receptor, the same guy measured by single particle tracking people. So this is the IMSD plot. If you zoom in, you can do all your whatever measurement and fitting to get the local diffusion coefficient. I want to stress that here below 1 millisecond, we have a bunch of points, so testifying the high temporal resolution. But most importantly, if you analyze quantitatively the shape of this curve, that is transient confinement, you get as well an estimate of the mesh size that now is much, much closer to what expected based on transmission electron microscopy, 130 nanometers. And you have an advantage that you can simulate, you can reproduce Kusumi or single particle tracking standard acquisition, because you can decide, okay, let's put our acquisition at 10 millisecond temporal resolution. Let's recalculate our IMSD plot at 10 milliseconds. Well, you realize that you get the same numbers of single particle tracking people. This is the new curve. You get a mesh of 500 nanometers. Just because you are averaging together many 100 nanometer mesh in a bigger one, just because you're going lower, okay? So this is a property of the technique, and you can compare data from IMSD with data from single particle tracking. So I want to stress this concept because in the field, you could do that variation FCS if you want an alternative to single particle tracking, so by increasing the spot size, or by decreasing the spot size on the other side by stead effect, so by stead FCS. In both cases, you get a nice view of the displacement, the diffusion law of the molecules, with the difference that in the case of stead, you can claim, the authors of this famous paper, 2009, by Egling and co-workers do, you can claim the direct observation of the nanodynamics because you are going nano and you see nano, the diffusion of the molecules. Well, somehow we are doing the same. I will claim the same in a much easier, let's say, measurement because we are working with a standard diffraction limited setup. And to prove that, to convince you about that, I can compare data from stead FCS with data from IMSD in a standard confocal microscope. This is a plot by the group of Stefanel. You can find on the web, not published, but you can find easily. Transferrin receptor, again, measure a different spot size that you see on the x-axis, and with the calculation of the diffusion coefficient. And you see, a threshold at about 100 nanometers, in keeping with what I show you so far, 130 average size of the mesh at about 100 nanometers, the diffusion coefficient collapse. Okay, that's so far so good. I can recalculate again my IMSD plot to put it on top of this data, just changing the y and the x-axis with these new coordinates. Okay, I'm putting my IMSD plot on top of this data. And if I do that, you see the coincidence between the two, with the advantage that they are superimposed, so you get the same conclusion, with the advantage that we're using a standard confocal setup, as I mentioned, and we have even more data points because we are not obliged to be stuck on the whatever 3, 4, 6 observation spot able to produce with a step microscope, but we have many data in a single measurement. So I hope I show you this potentiality of the technique. And the very last part of the talk, I want to mention that more recently we moved from the scale of the molecules, new peculiar, not new because it's known since a lot of time, but a new scale to apply IMSD, that is that of the subcellular organelles, okay, vesicles, transport vesicles, whose importance was recognized also by a recent Nobel Prize for physiology and medicine. These objects are, in fact, involved in many processes and in their pathophysiology, from neurotransmission to homeostasis, from signaling to metabolism. If you want molecular information from these objects, it's better if you refer to the talk by Alex and you do 3D orbital tracking to get molecular information. Still, if you want information on the object as a whole, you can also apply standard imaging and IMSD analysis, with the advantage that here you will get diffusivity, anomalous coefficient, but also information on the size, because in this case, we are not dealing with point-like particles, but with entire organelles, with a size comparable or even bigger than that of your observational volume. So we combine this time 3 numbers: estimate of the size, diffusivity, anomalous coefficient, in a parametric space in which each point has these 3 coordinates. Each point is an acquisition. And so you can easily get a cluster of points whose dispersion in space reflects the variability of your cells, of your acquisitions. And you can easily get, in few hours of experiments, an IMSD-based fingerprint of your dynamic nanostructure. And you can use this fingerprint to evaluate different cell lines, different experimental conditions, different metabolic states, drug treatments, and so on and so forth, with the advantages of IMSD I already stressed before. So we applied this tool to the fingerprinting of many intracellular substructures, late endosome, early endosome, lysosome, cluttering coated pits, macropinosome, caveolae, whatever you want, in a proof of principle demonstration of the validity of the approach. And then we focused on a secretory vesicle, very famous and popular, the insulin secretory granule, just because it's involved in diabetes physiopathology. And you know the beta cells is evolved to respond to glucose stimulation with insulin secretion, and insulin secretion is mediated by the insulin granule. Well, the last 2 minutes, I'm showing you data on the insulin granule labeled with insulin, a fragment of insulin to be precise, the C-peptide tagged to GFP, and we use that standard way of labeling the granule to get our fingerprint. You see on the left in green, the validation to be sure it was reflecting the behavior of the granules. And then we tested many different experimental conditions. One I want to mention is labeling itself. If you label the granule with a different protein, you get a different granule. And you can recognize very easily that by fingerprinting in a few hours, I want to stress, by IMSD analysis. In this case, on the right, the green is the fingerprint labeling the C-peptide, and the red is the fingerprint labeling a transmembrane protein. And you get two different pictures, the same object. And the literature is based on 50-50 papers labeling either way the granule. This is a message of also taking with a grain of salt in mind the data on the basis of these evidences. I'm going to close because I think I'm running out of time. Just to mention that these fingerprinting can be used now in many different fields at the subcellular scales to establish connection quantitatively between endocytic vesicles and cancer, differentiation. And also I want to mention membraneless organelles. They are very popular nowadays. They are actually very subtle and transient objects, but once we solve the way to label them and to look at them, these kind of techniques can be useful. And last but not least, the take-home message. I hope I delivered that the IMSD, as any other spatiotemporal SCS methods, is fast, robust, and provides information in presence of many molecules with single molecule, single object sensitivity, as any other SCS techniques. Is compatible with any way of probing space and time in a microscope, images, lines, orbits, whatever you want. Can be applied to multi-channel acquisition, just to have multiple readouts, but also to achieve cross analysis, cross in terms of, cross FCS, FRET, whatever you want. And from my point of view, is a step towards the way of filling the gap between standard spatiotemporal analysis and single particle tracking. And in this direction, many other techniques are doing the same job. I want just to mention the pair correlation going on the same aim, same goal. So with this, I conclude, and I just want to mention people in my lab, funding, and obviously special thanks to Enrico, because these data, these kind of activity in my career are always linked to the collaboration with Enrico and the LF people. I thank you for inviting me, and please ask questions.
Thank you very much, Francesco. Wonderful talk. Now we have time for some questions. There is one from Felipe Montesinos. He say, can it be applied on a TIRF microscope?
Yeah, sure. The data on the transferrin receptor were acquired in the microscope. If you are using a 2D system, the best. And each point at the same time, like you do in a camera-based.
We lost you a little bit, Francesco.
Sorry, can you hear me?
It got broke, the communication.
Can you hear me now?
Yes, we can hear you now.
Okay, sorry. Okay, if you can hear me now, I repeat my answer. No problem using TIRF. Actually, the data I show you on the transferrin receptor were acquired at TIRF microscope. So actually, it's the best option you have if you want to study 2D system, as you know. So you can apply this technique with no problems.
Excellent. Thank you. Let me see if there are any other questions or someone raising the hand. If not, I will thank you again, Francesco. And I will ask Enzo to share your screen. And I will introduce your next talk.
Thank you.
Thank you very much. So next talk is given by Lorenzo Scipioni. I already introduced him, but I will do it again. Lorenzo is postdoc at the Laboratory for Fluorescence Dynamics in the Biomedical Engineering Department at the University of California. And in this talk, he's gonna explain a method that he developed during his PhD. He was visiting the LFD and he was using the Airyscan detector that comes in any, well, not any, if you order, 880 and 980 microscope. It will cost you a little bit more, but it came by default if you have the money. But using in a completely different way that the reason for why size design it. And I found this a very nice application, and I think he will explain us how that's work and what are the things that we can do. Thank you, Lorenzo.
Thank you, Lionel. As Lionel mentioned, I'm gonna tell you about this paper we published, I think now a year and a half ago, 2 years ago, about how to apply correlation techniques, ideally all of them, to by using a detector array, in this case, the Airyscan detector. So starting by the motivations of this is that correlation techniques give you a lot of information on different spatiotemporal scale that are also complementary with each other. So some techniques can report on, as Francesco just described, on the environment organization. Other techniques can report on the particle dynamics, so on and so forth, like oligomerization state in case of number and brightness. So, however, they have their limitations. So like IMSD that was just described is the techniques that allow you to determine the diffusion modality and the environment organization at spatial scales below the resolution limit, right? However, in order to gather this information, you have to sample over a very large, a relatively large area, right? So, which is typically several times higher than the resolution limit, because you have to sample the diffusion of molecule across space. On the opposite side of this, we have FCS. So FCS doesn't really allow, it doesn't have the same potential as IMSD to assess the heterogeneity of motion within the confocal volume, but it's applied to a confocal volume. So it reports on a volume which is approximately the resolution limit. So what do we want to do? We want to get only the best, of course, of these two technologies. So we want diffusion modality determination, and we want it at the resolution limit and possibly below the resolution limit. So this is our goal. So let's go take a look at how things work. So when you have a diffusion process, you have your illumination volume, and molecules go in and out of your illumination volume, giving you a sort of spike of fluorescence, which give rise to fluctuations that you can analyze through the autocorrelation function, get your FCS curve, and obtain information about the diffusion coefficient, concentration, oligomerization state. This is very nice, very straightforward in when you are in a solution. But what happens if instead of a solution, you have a diffusion through obstacles? Obstacles that may be represented by either molecular crowding or a structure, more or less complex. What you get from FCS is just a diffusion coefficient which is slower than the diffusion in water, which is expected. It's what you get in cells, but it doesn't report on what is actually happening inside your illumination volume. One technique that was mentioned in the previous talk by Francesco is spot variation FCS. So the idea of spot variation FCS is to discriminate between several kinds of common diffusion behavior that can happen in the cell. So free diffusion, impermeable obstacles, dynamic partition, and meshwork. These kinds of diffusion give rise to these different curves, which I'll show you in a minute how to obtain those. And you can see that like meshwork diffusion has a very steep slope. Microdomain has a different slope, which actually intercept in the positive values in this axis. Free diffusion intercepting 0, and so on and so forth. So you see that these curves are what? It's apparent diffusion time over a confinement parameter, which is proportional to the waist squared of your illumination volume, right? So to construct these curves, you need several illumination volumes. So if you take a very small illumination volume, say smaller than the meshwork, than the size of the mesh, then inside that volume, the molecules will largely diffuse as free diffusion, right? So you will get a point here along the free diffusion line. Increasing the volume, you will get a higher contribution of the interaction with the meshwork. So you will no longer walk along the free diffusion curve, but you'll start needing more time, basically, to cross the illumination volume, right? If you increase even more the size of your volume, you will continue seeing this effect, and then you can extrapolate that you're actually walking around the meshwork with this size. If we had more points, we could actually trace this whole curve and obtain the exact shape of the structure surrounding your molecule. This is one thing. IMSD works on similar principles, but exploits the temporal dimension. So if you are in a solution, if you consider the time and how much is the broadening of the diffusion probability distribution, it will increase linearly regardless the temporal distance, right? Because there's no obstacle in this. In presence of obstacles, short times means short length, so that means that the probe is not, it didn't travel enough to actually interact with the structure. So you will have the same slope as free diffusion. At longer times, it will report on the time it takes for the probe to cross all this structure, and then you will have some transition in between. Per correlation function, this is a sort of summary of everything. Per correlation function tells you how the points around the points that you illuminate are connected with each other, right? So in a solution, there will be no delay between diffusion from one point to another. If you have an impenetrable obstacle, you will see connection only, in this case, in the vertical direction and not across the obstacle. If you have a barrier here, in this case, you won't see any connection between these points and these points. You will see a slower connection between these two because it has to go around. And so this one can report on the connectivity of the surrounding and the anisotropy of the motion, like directionality and so on. If you remember from the previous lectures, all these techniques are use different instrumentation. So you can go from like single point, photon counting detectors, cameras, and so on and so forth. Because some techniques need a fine spatial sampling, other a very fast temporal sampling, and so on. So you always have to compromise. With the Airyscan, you don't have to compromise anymore. This is because it is a detector array, so it has a spatial extension, basically, and it also can acquire with a very fast acquisition speed in the order of microseconds. So you can go 1,000 times faster than a camera, still having camera capabilities. Also, the primary purpose of using the Airyscan is to use super resolution. In fact, we can actually use all of the correlation techniques that I just introduced at the super resolution scale, approximately 140 nanometers. As a fast comparison, I don't want to go to basically, with the Airyscan, it's possible to do FCS, obtain the diffusion law at the same time in only one shot. It's possible to obtain the IMSD, PCF, and connectivity maps. Let's see how. So spot variation usually is applied by either like changing the objective using a lower numerical aperture. So it basically goes from your diffraction limit, and then you sort of increase the size of your illumination volume to get the points, get further points in your curve for the determination of the diffusion. In this case, what we are doing is we are just doing it post-processing, considering a variable number of detectors in our image. Since the detector, this is the reason why you can go in super resolution with this, is placed in place of the pinhole, it acts like a virtual pinhole that you can change post-processing. So if you take the signal coming from all the detector, it's like having a pinhole of 1.2 Airy units. If you consider a more central area, then it will be like 0.8 Airy units. So the points per function will be narrower. And then you can go 0.6, and then you can go 0.2. This will give you the higher resolution, so the smaller illumination volume. You can see that the FCS curves obtained by this volume, and then smaller and smaller and smaller volume, you can see that you have the G0 increasing, meaning that you are measuring less molecules, and this is due to the shrinking of this illumination volume. So this is what we want to achieve. So we have simultaneously 4 illumination volumes from which we can extract information about the diffusion law. These are the values of the volumes we have access to, and generally these are the 3 types of environment that can hinder the diffusion. So if we have free diffusion, the interpolation, the linear interpolation of these 4 points will cross around 0. That means that it's undergoing free diffusion. If it's a meshwork, it will cross below 0. If it's a macrodomain-like, it will cross above 0. One other thing that we added is looking at the number as a function of the effective volume. This will report also on aggregation and exclusion of, so if you're looking at an area which excludes the molecules, or if it's a sort of domain in which the molecules are more accumulated. This allows us to create this 2D space in which if we have a point in this black point, for instance, it will be a region in will mean that your probe is undergoing diffusion in microdomains and is excluded by the surroundings. So there's going to be a portion of the illumination volume which is not accessible to the probe. This is orthogonal, so you can have like meshwork with exclusion and aggregation, microdomains with exclusion and aggregation. And the farther the points are from the origin of this space, the higher is the effect. So the higher the exclusion, the higher the aggregation, the higher the confinement in these microdomains or in the meshwork. IMSD can be also done across the detectors, and then it can report on diffusion, free diffusion, directed motion, confinement, and so on. Percorrelation can be done by doing the percorrelation between the central detector with the ring at a distance of approximately 50 nanometers. This other ring, another ring, another ring. We're looking at how the shape of the constant of the percorrelation function is modified to assess the directionality of the molecule in that super resolution volume. Also, you can look at the percorrelation between each of the detectors that you have available. So if you consider the red detector and then this blue group of detector, or this, or this, or this, and you look at the cross correlation, the percorrelation between the black and any of these, you will get the typical curves that you are used to when you look at percorrelation functions. So the farther you go in space, the longer it takes for the molecule to reach them. But this, the distance of this one is subdiffraction. You are in the order of tens of nanometers, right? So you have a much higher sensitivity than the common percorrelation techniques. And also you can use all the cross correlation between the detectors to create a connectivity. So how does a connectivity map look like? This is in case of a solution. You can see that it's everything's perfectly isotropic. The diffusion has the same value in every point of the detector. The thing change as soon as you move to the cell. So let's see this data set which was acquired in a cell. And here we have the connectivity map, and here we have the anisotropic. So one thing that you can notice is that you have an area here in which the connections are slower than the rest of the region. And if you look at the anisotropy, this region corresponds to an isotropic area, whereas the surroundings, the molecules tend to avoid this region, right? So what it means that you're looking at an obstacle to diffusion at the nanometer scale in which the diffusion inside the object is relatively isotropic and the molecules tend to flow basically around it. So specifically, this is a region of chromatin compaction that I've been analyzing, and that means that it had a region within that heterochromatin region which doesn't allow the molecules to flow in. So to show you an application of this, let's look at some general cellular compartments. So what we call unstructured cytoplasm. So it's typically on the edges of the cell where there are not many organelles present. The structured cytoplasm is what you see closer to the nucleus, so where you have all the other organelles creating some sort of crowding. So you have like the mitochondria, microtubules, and everything that's usually relatively closer to the nucleus. Inside the nucleus, we have heterochromatic region, euchromatic region, and the nucleoli. So nucleoli are typically very dense, and heterochromatic regions are compacted DNA, so they should be denser than the euchromatic region, which are typically decompacted DNA. So let's see where these points fall in our 2D plot. And you can see here that the unstructured cytoplasm show a little bit of confinement. So they all fall in the region of exclusion and microdomains, like diffusion, right? But the unstructured cytoplasm and the structured cytoplasm have roughly the same confinement in terms of microdomains, but they have the structured cytoplasm has a higher exclusion. So meaning that the GFP, in this case, which is the program we're using, does not have access to certain organelles, which is known because they're membrane bound, so it's not complicated. You can see also from the image that you have darker region in which the probe can't diffuse it. As soon as you move to the nucleus, everything is shifted toward higher exclusion. As you can see here, these 3 points are relative to the nucleus. This is the heterochromatin. So the heterochromatin has a confinement which is relatively similar to the cytoplasm, which is also what you would get if you were measuring simply the diffusion coefficient, right? The diffusion coefficient of GFP in the nucleus and the cytoplasm is largely similar. But as soon as you move to heterochromatic region or the nucleus, you have a much stronger confinement in microdomains. So this all is telling you a nanoscale picture of what is happening inside the nucleus. And on top of this, all of this is done with minimal interference with the sample because we are not labeling anything with anything. We're just using GFP, which is an inert probe, which doesn't interact with the cellular machinery, to report on the structure of the surrounding. We can also do IMSD, and we can obtain diffusion coefficients. We can obtain length of confinement, and so on and so forth. We could, in principle, also report on oligomerization states, concentration, environment organization, connectivity, anisotropy, diffusion modality, diffusion coefficient, all in one single point measurement of approximately 30 seconds. So this has been published exactly like a couple of years ago. And now what we'll be doing next is create another microscope which relies on another detector to include also single photon counting detection and lifetime measurements or hyperspectral capabilities, for instance. And that's it.
Thank you, Lorenzo. Excellent. Thank you very much for the presentation and to illustrate us how we can obtain unbelievable information from a device that was conceived for a completely different objective. For those of you who don't know about the ID detector, it was constructed in order to do super resolution. And I remember when Lorenzo started with this work, he had to do unbelievable calibration in order to figure it out, all the crosstalk for every single detector, all the calibration. And I spent hours and hours discussing with him about how he did to analyze all these issues. So
it's a terrible instrument. The electronics of that system is.
So there is a question or two. I don't know, from the audience. Also, the people can ask. I have a stupid question, Lorenzo. I remember how difficult it was to be sure that you are in the point, and after you measure, you are again in the point. Kind of similar of the orbital tracking. In the new instrument that you are designing, there is any chance that you can use this kind of approach in order to be sure that you are not missing the point where you want to be, if your object is has some constraints in the volume that you are analyzing.
Yeah, so technically the Airy detector is a sort of micro camera. So even though the number of pixels is limited, it's like you are actually looking, you have an eye on the point spread function, right? So that can also be used, for instance, you can have one color with which you can track your structure, or you can see, I mean, in this case, if the objects are dimmer than the surrounding, you can use that information to sort of actively track it. And you can actually use it for orbital tracking, for instance, right? Because it is effectively actually a camera, and you can actually do orbital tracking with microsecond resolution, for instance. Yeah. Or stuff like this. But yeah, I'm also working on trying to figure out how to have an active feedback between the images and the stage of the microscope in order to constantly correct for motion artifacts. With sometimes, especially for correlation, and sometimes you get stuff and you have to throw away the measurements because something moved.
There is a question from Amit who says, can this technique be used to find local distribution of oligomers and the cell, since it can evaluate anisotropy, or it's just an average value for per the cell as in SIMFCS?
I'm not sure I follow. So the oligomerization state you can obtain from number and brightness, or if you have some a priori knowledge, there's also this enhanced number and brightness, which can also infer on the actual distribution of oligomers, for instance. That can definitely be applied to this. I don't recommend it because, as I said, the electronics is terrible on this system. But if you had a true single photon detector, that would be awesome, actually, and relatively easy to do. So you could also obtain that information.
I think the question of Amit is more regarding that if this is just an FCS value, and it's not, because you have 32 FCS trace for each of the detector who has special information that you don't have in a single FCS. In the single FCS, you have the whole points per function in a single point. Now you are taking information, the points per function for different location. You have distance of few hundred of nanometers, right? Because each of the actuators,
yes.
the nanometer, Lorenzo, right?
Yeah, so in general, so this can probably be a more general answer that actually applies to all the other techniques as well. So this was thought as a method to use the FCS concept to probe the organization of the environment. So why is that? Because typically you have to, your measurement time is usually in the tens of seconds, right? So if you do the calculations, the typical diffusion in the cytoplasm is in the order like ranging from microseconds to tens of microseconds, microsquare per second. So by the time you start and finish your measurements, you have constantly new and new molecules coming through your illumination volume. So also molecules that were completely on the other side of the cell, by the time you finish your measurement, they will have diffused back and forth several times, right? So technically, you're always averaging. You're always averaging. What does not change is the region in which you measure. That's why it's the important information that you get from these techniques is the local information. Or compartment information. For instance, in the nucleus, may be different from the cytoplasm because you can have, so if the molecules are big, you don't have passive transport. You need to have an active transport. And then
to add something that I don't know if it was clear from your presentation, that you do only one measurement and you obtain all the parameters that you show. So it's the same measure. And that answers another question that Sebastian Acuna has. How easy is to get the raw data? Well, if you know how to get it, it's very easy. Each channel has a separate sequence, but you need to know how to read it. And if you want, we can give you the routine for reading.
Yeah, okay. It's less stupid than you think. Sebastian.
No, either you know or you don't, or there is no way you can guess it.
Yeah.
And the other thing, exactly. The other important things to say is that Enrico and Lorenzo produce a particular software, which is a version of CMFCS for Airyscan, who is available at the LFT, and you can do all these calculations that Lorenzo was showing in the presentation.
Yeah.
Yes, sir. I ask because last year I heard that in the old Airyscan microscope, it was possible through some bugs in the software, but in the newest one, it was difficult. That was what I heard. So that's why I was asking.
I'm not familiar with the new one. So you can from the software you can actually look at the images themselves. But if according to how you save them, for instance, I was saving them in OMETIF format, and then it saves one file for each of the channels.
We read the CCI format, and the CCI format has 64 channels total, of which 32 are the direct information. Feature channels
Super
are
weird.
And the other are images which have been processed. So either you know which one you have to read, or you have to wait some time to figure it out which one. And I don't believe they changed the CCI format. So there is, we have in TIFF because we convert in TIFF, but the original signal is only CCI, and there is no reader that I know of any software that will give you the information of the single detector. If you have a new instrument, send us the data, and I will tell you if I can read or not. Okay.
Okay. Thanks.
Okay, so let's move to the next talk, which is the last of the day. And this talk will be given. Suman, I think you are able now to share and open your microphone. The talk will be given by Suman Rashid, as I introduced him yesterday, he's assistant professor at the Department of Biochemistry, Molecular and Cell Biology at the Georgetown University. And today he's going to talk about application of the driver microscope. As I introduced him yesterday, he's perhaps the person who took more advantage in the world about this microscope and is the right person to show how all the possibilities that this microscope offer. So thank you, Suman, and I pass the word to you.
Thanks. Can you see my screen and hear me properly?
Perfect.
All right. So yeah, this is, of course, it's going to be more of an application talk, and I'm going to talk about some of the stuff that I did, along with some of the stuff that Lionel did, and but the common theme is, of course, the use of the diver detector. And I'm going to, I didn't look at it, but I'm going to guess you already have been told how the diver detector works, but I'll just go very briefly and then move on to the actual applications. So the diver detector, and this is the picture that is assembled in Georgetown, not the one from LFD, but it's basically the part which is right here. So it's a conventional 2-photon upright microscope, which is from Olympus, which is shown here, and the diver detector is here. The diver detector is a it's in the forward direction, the imaging is done. And basically there's a refractive index matching between the sample, the detector assembly, and the PMT, which stops the loss of light due to reflections. And this is sort of a blown up version of it. So inside this, you have multiple filters that you can use a stepper motor to move into, and that lets us image the different things. These are all glass filters, and now there are 2 filters which can let us do multispectral imaging using sine and cosine filter, similar to what Nick talked yesterday. And so when you extract the sample in a conventional microscope, the objective itself collects the detection, but in diver, the wide area photocathode, which is 1 inch by 1 inch, so it's a much larger area that collects the light. And as you go deeper and deeper, this angle actually increases, which gives the diver is increased sensitivity at a deeper level compared to a normal microscope. There's also an effect from 2-photon, which has been explained by Chris Joe before, where the normal microscope looks less just below the surface. So this gives the diver a very high ability to look deeper into the tissue. And the refractive index mismatch is a very simple idea that you can see if you put a bunch of glass slides and look through the image, some writing LFD here, you see a very faint, but if you put just water in the image, which is not a perfect refractive index along to glass, but it's a better mismatch from 1.1 to 1.52. And here is 1.33 to 1.52, and you see it much brighter. So that's the basic idea of, and due to this loss of reflection. And the other important part is because you do not have a lot of optics here, light, especially in the lower wavelength range, UV and close to a very low visible, which in a normal microscope, by going through the different optics, gets lost. You don't lose that. What that means we can use the full fold of a laser and do second and third harmonic imaging with a normal, here it's inside, but even a titanium sapphire microscope. Because if you do third harmonic imaging with a titanium sapphire at 1050, the wavelength that you will see the light is at 350, which is very difficult to see in a normal microscope. So you get up to 10,000, up to 1,000 times signal gain based on the situation. Now, the difference is, so what I'm going to talk about, a part of that talk is going to focus on harmonic generation microscopy. So I just wanted to introduce that. So in fluorescence, you excite the molecule. So this is 2-photon excitation. It goes to excited state, comes back, and the emission happens basically all angles around that point of excitation. So you can pick up from any angle. However, in second harmonic generation, what happens? You basically send an oscillating field, but because of the breakdown of the center of symmetry of the molecule, for example, collagen and myosin in the tissue, the electronic oscillation here goes equal height in both directions as you are putting a field. However, it goes, there is a resultant field because this is an anharmonic potential. And that gives rise to the second harmonic. Now in second harmonic, the most of the light goes in the forward direction, and only a very part of the light gets reflected back to the direction of the excitation, and that's the normal conventional microscope way. Again, it goes into a state which is not exactly a proper excitation state, and because it's a virtual state, there is no delay associated with it. So in fluorescence, you go to the excited state, you relax and then come back, that gives you this lifetime of fluorescence. However, in second harmonic, because it's a state which is virtual, there is no delay and you get a lifetime of 0. Now, in terms of excitation, you can excite anywhere from 700 to 800 nanometer, let's say, and you get a signal which is second harmonic at 350 nanometer if you excite it around here. And you get the fluorescence signal which is normally coming from tissue around 400 nanometer upwards. By doing that, you can separate the second harmonic very easily from the fluorescence signal of the system, while doing the same excitation at 700 to 740 nanometer for both. So you can use one-shot excitation. Now, this is just to show how the second harmonic images look like. This is fluorescence, this is in kidney. So this is basically looking at collagen 1 and fibrosis, and I'll come back to it in a second. Now, as I mentioned, the diver is in the forward detection, and as you can imagine that you'll see much more second harmonic signal in the forward detection. So by looking at the diver detector, which gives with its high sensitivity, originally, although it was developed for deep tissue imaging, we figured that it would be really good for imaging harmonic generation, and that's sort of what we did. The other important part, again, as I mentioned, this is actually excited state, but this is a virtual state. So the diver, the signal of SHG from the diver will resemble the laser pulse. However, there will be a delay in the fluorescence signal. What it means in terms of phasor is your fluorescence signal would be somewhere in the middle, but your second harmonic signal would be basically at this position, which is lifetime of 0. And that gives you another separation from separating out bleed-through fluorescence or anything else that might combine the complicate the second harmonic generation when excited at 700 to 750 nanometer wavelength. Now, the first thing we use this is we use it to separate collagen types, originally based on flame. So these are just intensity images of collagen gels that was formed in the from solution in collagen 1 through 5. And when we actually used the phasor lifetime to see if they have different lifetime, they seem to have different lifetime, especially collagen 1 and collagen 3. There's a large separation, and these two are important because they are involved in different types of fibrosis. Collagen and 4 and 5, they don't really form fiber-like structure, that's why you don't see them. Now, when you take the second harmonic imaging, you can do basically the same area. So you can do second harmonic imaging. So this is the intensity. However, we can use now this phasor to see, okay, if it's actually second harmonic or is something else. So it turns out that collagen 3, the signal is mostly coming from fluorescence and not second harmonic, although mostly because this is so bright compared to the other collagens that is bleeding through the collagen second harmonic signal when excited at 750. We use this even in combination of where we mixed collagen 1 and 3. So this and at high fluorescence range, which is the lifetime of collagen 3, you can select out collagen 3, and low fluorescence intensity range, you can select out collagen 1, and you can see the second harmonic is all collagen 1 and non-collagen 3. Now, we use this idea to look at mouse femurs So this is a whole bone section of a mouse femur. Now, one of the things with the diver is it lets you image really large areas. So I think right now it lets you image up to 1.2 millimeter to and 1.2 millimeter. That's the field of view, which is pretty unheard of in normal microscope. So by doing that, we can stick the autofluorescence and second harmonic image, and we can use their lifetime behavior to show that where the collagen 1 goes, where mostly collagen 3 rich areas goes. Now, of course, there is a overlap here between the NADH, which makes this analysis slightly more difficult than if it was just collagens, right? But we could figure out a way on a particular type of mice, which is called smart MRI data. It's a mice that develops bone myelofibrosis. And we could see the extent of the myelofibrosis by basically using a color coding between collagen 1 and collagen 3 to see how collagen 3 increases in the internal part of this bone on this smart MRI device. So that was my first work with the diver imaging system, and that was published in 2015. Now, one of the other main projects we looked at is quantification of the UBA mice model using Freeman SAG. So let me explain the model first. So what you do is you tie up one of the kidneys, and as the blood flow is restricted, it basically becomes fibrotic over time. So you take, once this is done at different time points, you sacrifice the mice, and you look at, you slice the kidney, and you look at the amount of fiber generation over time. And again, we are using green color here to separate out the second harmonic and red color to separate out the autofluorescence that is bleeding through to the second harmonic channel. And if you look at 7 days between the control and the disease mice, you see there's a large difference already started to show up in the fibrosis or collagen 1 accumulation, especially around the glomerular space, which is important for this sort of kidney fibrosis models, right? And then at 14 days, it becomes worse compared to the control. And at 21 days, it goes really, really bad. And then we can quantify this. We can quantify basically how many pixels are covered by the green compared to how many pixels covered by the autofluorescence. And we can see that ratio actually increasing with time. Now, one thing to be noted here is the right kidney, which is actually is not getting, is normal, that's also getting worse because it's doing 2 kidneys work by itself. And that hasn't been really shown in conventional stuff before, but we could see that. Now, another important part is like we can look at each individual sample. So there are 6 samples originally in each with 10 images in each. And each case with the days of UBA, you see this ratio going up in every single area of these mice. Now this y-axis is different for different ones. So of course, the time with time, this is a much higher, it's 5, and with 7 days is only at 0.4. But we can see that, we can quantify that. And we can, of course, because diver is a 3D imaging system, we can look at it, we can look at how these fibers are oriented in the 3D. Now, one other important part of using this sort of idea is this. This is both 7 day, same areas. This is, so this is all 7 day. Same areas imaged both times. This is with normally how people do it with background correction. If you do background correction, you're also going to lose fibers which are not exactly oriented along the path that you are exciting. And that's why you see a much less signal difference between these two. So we couldn't differentiate originally, but these samples are done without any background correction. And by doing that, because we are not getting rid of any signal, we actually get a much better separation between the disease and non-disease states. So other examples, so this is basically Lionel and Sasha's paper, with Enrico. So they have looked at mouse brain, which has been labeled with YFP. They can go up very deep. And these are mouse brains which are non-clear, I believe. So most of the time you see these images, they're in clear tissue, but this is in a non-clear tissue. They looked at high-fat mouse liver in 3D. Again, they can look at and separate different things. Again, this is an eye of a mouse embryo, and if you can select different lifetimes, different components, and I believe so this shows up how different components are oriented in a mouse eye. Then again, in the other part of the paper, Lionel's, because he is the guy who does all the lung stuff, they have looked into rat lung to see the separation again in there. And these are again large area images, so it's not very difficult to do. And you can see the difference between two different types of lung. And if you have questions about that, I'll just refer to Lionel about that. The other examples, of course, is third harmonic generation, so I didn't go into the detail of it, but third harmonic generation is basically the way you compare. So third harmonic generation happens at the age of a diffractive index mismatch. So if you have two separate diffractive indexes which are very different, it will be a high mismatch and it will give a really high third harmonic generation separation. To do that, we excite at 1050 and we look at 350 nanometer, same as SHG window, but the excitation is different. And by doing that, you can see this is the fluorescence image and this is the third harmonic image. And you can see in adipose tissue, you can see the tissue boundaries very well in third harmonic image. Fat droplets, which are big and have a very different refractive index than the rest of the image, that can be easily seen. So this is here what is seen here. So these are low fat and high fat diet. And in high fat diet, you can see the fat droplet and the age of the fat droplet being really bright. You can also look at things in the cells. I believe these are again droplets, and in the nerve, and in bacteria, because again, you can do basically image of the bacteria, non-labeled image of the bacteria using the third harmonic generation. With a sine cosine filter that has been explained before, you can again use a sine cosine filter. And these are the same bone samples I originally showed. So this is different, again, lifetime separation. And this is the lifetime phasor image. But as you can see, with the spectral phasor that is collected with sine cosine filters approach, you can separate out the collagen 1 and collagen 3 areas exactly how you would separate out in lifetime. So in this case, they are complementary, but you can imagine by combining these things together, you get much better separation between things which has either overlapping spectral information or overlapping lifetime information. So you'll be able to split them out into more. Now, another thing that we used in, and this is slightly changing the time, slightly changing the field, is human kidney biopsies. And this has renal parenchymal injuries. So they basically, from patients of diabetic nephropathy, and they're looking at what's the injury associated with it. So when we originally started, we basically took the images, and these are different extent of image. So this has no disease, it's normal. And then we have mild, moderate, and severe diseases, and these are core biopsies, right? And if you look at the phasor plot, you see that with higher disease, the phasor goes to higher phase angle. So the lifetime is actually increasing as the extent of disease is progressing. Now, we wanted to quantify that, and the first thing we did is the original, how phasor has been analyzed for a long time, is by splitting this histogram into two and seeing what fraction of one compared to the other. And as you can see, the MCD has mostly purple, and this one is mostly yellow, the severe one. And of course, we can do at different areas, different parts of the images. By looking at that, we could potentially calculate just by this fraction similar to the second harmonic analysis I did before. Doing that, we can separate out the MCD from the other disease states, but we could not separate out individual disease states very well. And the reason is very simple. There is a lot more variability in this system. So if you are averaging everything out and giving it one number, you are not going to be able to get the heterogeneity in the system to look at. And so these are just phasor plots. You can see the phasor plots moving. So we went back to something that I did with Enrico some time ago about which is called multi-parametric phasor approach. So this is an approach which is based on machine learning towards phasor analysis and is oriented towards separating stages of disease based on receiver operating characteristics curves and how we can separate them. So, and it's based on a method called distance method, which is originally developed by Enrico quite a few years ago before this. So the way it works is you have your phasor plot, but this is a heat map. And you can think of this as a 3D plot where the y-axis is basically the number, the z-axis is number of phasor points. The way we did it, we split it into 4 separate segments. And in each segment, we calculate a few different parameters. We calculated the center position of that particular segment, which is G and S, the length and the width of this that is fitted into this. And then you also can calculate the number of molecules and the angle it is creating with this 0, 0 point in the center. Now, so from each of them, you get 6 parameters, so 24 parameters if you split into 2. And those 24 parameters can allow us to separate between the samples. One of the ways to think about it, let's say you have only a few pixels, which is up here. So if you do average, you might not catch it. But if you split it into 4 at the lower intensity level or lower number of phasor points level, they will be actually important. So that's what gives it the sensitivity. So based on that, we calculate the average spectrum for the 2 separate groups. One is for the control and one is for the disease state. And just by putting this weight associated with each of those 24 parameters, and once we have that average spectrum, we select out how each of these spectrum is differentiated, and we calculate the maximum separation between the 2 groups, which is, again, the control and the most disease. And once the separations are done, and we know these values of WI, we can actually use that to create the separation index. So this separation index would define how a particular image goes into the disease state or in a non-disease state. So in terms of actual imaging, this is how it looks. So if they're very well separated, the 2 groups, they would be at -10 to +10. That's the number here. If they're not that well separated, it will be somewhere in the middle. But once the separation is achieved, you can use a unknown sample, which you don't know what the extent of the disease is, and you can select out where this appears. So this case, this is more towards control, this is more in middle, this is more towards the disease. But it can also happen that some areas of it are actually in the control, some areas of it in the disease, and this will let us separate each individual image compared to the whole sample by itself. So we did that. So we looked at the mild to separate. So we used MCD and severe to separate them out. And then we looked at mild and moderate. So this is all results together, and we start seeing the shift towards more and more disease states as disease is progressing. And this is just to show that we get a really good separation between the two types of state. And this is individual images. Again, in this case, you see some of the areas are on this side, some of the areas on the other side, but which is even more evident here. But in some cases, they are only disease, so only mostly disease. So we did this for mild, we can do this for moderate, we can do it for individual images. And by doing that, we can separate out which of the areas where it is that the disease is shown up. And that's sort of got published this year. And that's pretty much all I wanted to show, just some examples of the diver detector and how things work. And finally, of course, in LFD, everybody, this is the whole picture. I like it because this is one picture where Lionel and me are there in the actual picture and not just others. Of course, Enrico, without whose help and everything, nothing would have been possible. Again, the Ramon picture and the Belen and Alex, other people from the lab, Rupsa, Mika. And my collaborators here, some of the work has been done with Moshe Levy's lab, especially the ones about the fibrosis, and also people from University of Colorado in Denver who were part of the people who provided us with the mice. And Dave is here. That's pretty much it. You are muted.
Perfect.
So, Suman, thank you very much for the talk. Wonderful presentation, and you show us how to take advantage of this fantastic microscope. I hope soon we can have it functional here too. Time for question. You can raise the hand if you want me to mute, or you can also place any question in the chat.
I want just to add something to the presentation. So all the samples we have are just natural fluorescence of natural second harmonic generation. So you don't have any enhancer. You don't have any dye in those samples.
No, they're all autofluorescence or second harmonic or third harmonic. They're unlabeled samples. Yes.
So essentially, is whatever they come from the clinic or from the libraries that we have.
Or in the case of the sample, the neurons that Suman showed from the paper with Sasha. This was a collaborator of Sasha that produced these mice with the yellow fluorescent.
Well, that's another thing, yes.
That's the only one.
Completely in just fixed without any cleaning. So with all the background, with all the scatter that you have in a sample without any cleaning.
Yeah, and getting 900 micron inside even in a brain tissue without clearing.
Those of you also who work on clinical sample, you can work with paraffin included sample or also with frozen sample.
But indeed, I want to make a point. You remember when Simon was starting with the adaptive optics, one day I challenged him and I said, well, I think I have good sample for you. And it was just a stack of paraffin with a lung within it. And it was, I don't know, thick like a centimeter. And he did the image across the whole set of the lung. And we learned that basically the lung is probably one of the worst organ that you have to in the body to image, because you have sac of air and then tissue. So the change in the index of refraction is huge across the lung, and it's very, very difficult to image. So yeah, even though the diver can break this problem and you can do 3D stack easily.
Yeah, and most of the stuff I've shown today, because I didn't go into the long lifetime species and all the stuff that I did, I was mostly focusing on the harmonic generation part. They are all done in fixed samples and samples from the clinic, because a lot of them, basically, all the biopsy samples came from Chicago and then imaged them. So I do have the data on both frozen and fresh sample, frozen and paraffin samples on those, and instrumental wise, everything works on both conditions. So yes.
Okay, so there is no request for the breakout room. So if anyone has a question for any of the previous speaker, I encourage all of you to take the chance to discuss with him now. Otherwise, I will leave them, in particular Francesco, because it's very late in Italy. So it's your chance to ask question. So if not, well, we closed the third day. I am very happy and I'm very thankful for all the wonderful talk that we have today. All were a fantastic talk. about all the possibilities from super resolution, new instrumentations, and also this kind of application from pathologies, as well as the spectroscopy that Francesco showed us in order to do biophysics conclusion from different biological questions. I saw Felipe there. Maybe he has a question. Do you? Okay. Well, it's not. I wanna thanks again, and
Let's meet tomorrow, Thanksgiving.
Tomorrow is Thanksgiving, so yeah, I apologize for all of you who are in the other side, in particular in America, for this. It was funny because this was organized originally to have a physical meeting, and this is the perfect week to have Enrico and Dave here. But now we will be able to change. Unfortunately, we book the room and everything, and we organize everything here in Uruguay, and we got stick with this day, and we couldn't change it. So I apologize with those of you in America, and we will celebrate Thanksgiving here. I will not promise if we will have a turkey, but anyway.
We will have asado. That's what matters.
We had an asado at Leonel's last time I was there, so it was terrific.
Exactly. Last year we celebrate with an asado in Florencia Irigoyen house. Yes. Okay. Thank you very much. See all of you tomorrow. Bye.
Bye, everyone.
Bye.
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 Leonel, I'll stay on.
Yeah, please.
Oh, I was going to say something. I'll just stay for a second.
Ah, a smaller group. Leonel, you appeared twice.
Yeah. Let me turn off this camera because
You didn't turn off. You just switched on the microphone.
Sorry. Okay. I'm tired now. Discovered if I do not turn off the camera of my computer when I speak, I am not in the screen because it only projects the video of the people who is speaking, not any other TV video.
You have to set it up. Just update your Zoom. There is a setup because because you are the host, I can't do that. So there's a setup where you can not only set up speaker, you can actually use somebody to pin them. So let's say one person is talking about it, so that person would be pinned, so you can watch him while other people talk in the background. So that's another way. And the new Zoom has filters. You can put in like, I don't know, like mustache and stuff like that, the newest updates.
You know, Suman, Susanna commented to me that, of course, she said, I look much thinner, and she said, you look thinner too.
Yeah.
You've been losing weight too, Suman?
I agree.
Yeah, I lost 13 kilos since you saw me.
13?
It's close to 30 pounds now since
30.
you saw me.
Okay, well, you're not up to me. I've lost 75, but you're
yeah, you look small. I was going to say that yesterday, but I didn't want to say in front of other people. You look so much thinner. I'm not kidding.
Of course, actually, this pandemic has helped me because I've been stuck here at home for months and months and months. And every morning, except for this week, this has messed this up, but every morning I go up to the pool and I swim more than a mile every day. Yeah. And believe me, that's a good way to help you when you're losing weight.
I've been working out, and then because of the pandemic, I stopped eating out.